Hello, Sourav.
Yeah.
Shall we start?
Yeah, we can start.
Can you hear me? Am I audible?
Yes, I can hear you. Can you hear me?
I think some connection problems there. Your sound is breaking. Okay.
Uh, Let me see. I can hear you clearly.
Okay. Now it's okay. Now it's okay.
Is it better?
Yeah, better. Yeah, it's
Okay.
okay now. Shall we start?
Yes, I'll just share my slides.
Okay, I will say a brief intro about you. Then we can start this.
Okay, sure.
Okay. Yeah. Sourav Datta is currently an associate professor at the Department of Biological Sciences, IISER Bhopal. Dr. Sourav was a brilliant student and passed his master's studies with gold medal from Pondicherry University and got CSIR JRF during his master's. He got a fellowship and did his doctoral degree from Gothenburg University in Sweden and completed postdoctoral research from University of Oxford. He had won two prestigious awards from for postdoctoral studies. First one, EMBO long-term fellowship for postdoctoral research, availed in 2010. Second one, Marie Curie Intra-European Fellowship, IEF, for postdoctoral studies in 2008. He came back to India with Ramalingaswamy Fellowship from Department of Biotechnology, India in 2013 and joined IISER Bhopal as Assistant Professor of Biological Sciences. He established his plant cell and development biology laboratory in IISER Bhopal. Since then, he and his students did extraordinary studies and published breakthrough findings in frontline journals like Plant Physiology, the Plant Journal, Plant Cell and Environment, Nature Plants, Plant Cell, etc. With this short introduction, I would like to invite Dr. Sourav to join SAMEEKSHA webinar series organized by AKRSA KFRI. Dear Dr. Sourav, please take off from here.
Okay.
Sourav?
Are you able to see my screen?
Yeah.
Yes?
Yeah,
Okay.
it's okay.
Okay, so thanks a lot for the kind invitation, and especially Nafis. Nafis, I really, it was nice to get your mail, and then obviously I had to agree. So today I will. I'll just give you a brief introduction about what I'm going to talk about. Is it clear? I am hearing some background noise. Is it clear to all of you?
Sorry, it's rain. That's why some noise is coming.
Yeah.
It's
Hello?
Yeah, we can hear you.
Yeah?
So, yeah,
Okay.
you can start.
Okay. So just let me know at any point if you, hello? Yeah, at any point if you are not unable to connect to me, just let me know.
Oh, okay.
Okay?
Now it's okay.
Okay. Yeah, so thanks a lot for the invitation. So I am Sourav Datta, working at IISER Bhopal, and my lab, we are interested in understanding plant environment interactions. And today what I'm going to talk to you about is the identification and characterization of two molecular players that we identified, which play an important role in how the environment regulates plant development and defense protection. So before I start, just a small introduction about the lab. So we call our lab the Plant Cell and Developmental Biology Lab, PCDB. This is an image of the lab. Although it is pretty representative, I think my students might agree that this is probably for the photo. And in our lab, we are using the model plant Arabidopsis thaliana, which we use to understand how a plant responds to environment. And among the environmental factors that we are interested in, one is light, that how light regulates plant growth and development. And the other is about how nutrients in the soil, they affect root development and how we can enhance nutrient uptake. So today's talk will be about two stories, and both of these are related with the light aspect of the work in the lab. So let me tell you about the first story first. So the story one is actually a serendipitous finding in the lab. So let me give you a brief anecdote how this story started. So what you see here on the screen is a plate where we have put this Arabidopsis seeds and the seedlings have emerged. And on the two sides of the plate, you see that on one side there are some seedlings which look more white, they are kind of bleached. And the other side, the seedlings look quite green, so they are not yet bleached. So basically what was going on here was a very talented graduate student in the lab, Arpita Yadav, who has just finished her PhD now. She was working with, as I told you, we work with how light regulates, how the different light components, how they regulate plant growth and development. So she was working with one of these transcription factors called BBX31, BBOX31. Our research is we generate mutants in different genes. In this case, she had generated mutants of BBX31, both loss of function mutants and overexpressed BBX31. So plants which had enhanced levels of expression of BBX31, so 35S, using a 35S promoter. So these, she had generated these, and she was doing experiments with these transgenic plants. And she was putting them together with the control wild type plants, seeds in different light conditions. And in one of the experiments, she had popped this plate inside a UV chamber to check what effect when you change the expression of this gene BBX31, what effect it has on plant development under UV. And she was supposed to take it out after a week, and for some reason she forgot about that plate, and she took it out after 3 weeks. And when she took it out, then this is the plate which we see, which I'm showing you here, is what it looked like after 3 weeks, where you see that all the col 0 seedlings, which is the wild type, they had bleached. Most of them had bleached under UV, whereas this overexpressor of BBX31, they were quite green even after 3 weeks of UV treatment. So she brought this plate to me and she told me that this is what happened and this is what she saw. So I asked her to repeat, and then once we repeated the experiments, we saw that every time we were seeing similar effects. So we wondered that what is this gene doing, that it is in a way giving protection to the plant against UVB. So the question we asked was what role this gene might be playing to protect plants from UV radiation. So a bit of background about UV. So this is the solar spectrum, and you see the visible light in 400 to 700 nanometer. And then you have the ultraviolet light, which can be divided into UV A, B, and C based on their wavelength. And the UVC, which is the most high energy UV, fortunately that is blocked by the ozone layer. But UVA and B, they can penetrate and they can reach the Earth's surface. And among this, UVB is the one which has tremendous impact on human and plant health. So, in humans, you probably know about the effects of UV, like skin cancer, cataract, these different kinds of, if you're exposed to high doses of UV, you can have these kinds of effects. Similarly, in plants, the UV radiation can have different kinds of effects. And these effects are totally dependent on the strength or the dose of UV. So what I mean here is that if the dose of UV is very high, then it acts as a stress response, and it can damage biomolecules like DNA proteins. It can generate reactive oxygen species. Whereas if it is low, then in fact, it can be a bit kind of a positive factor in plant growth and development. So it can modulate the growth and development of plants in low doses. And this, in fact, is also utilized in the production of several secondary metabolites in plants. So you must be knowing about different kinds of secondary metabolites in medicinal plants, phenolics. So all these can get enriched under low doses of UV. And this is being utilized in different industries, to enhance flavor and different kinds of, wherever you have importance of the secondary metabolites. Now, a bit about what we know about how this UV light is sensed by the plant and what is the molecular network that is known to us. So UV, hello? Hello, can you hear me?
Yeah.
Okay. So UV light is perceived by a photoreceptor which is known as UVR8, which is in dimeric form, and then upon UV, when it perceives UV, it monomerizes, and then it will bind to COP1, and this complex is involved in a signal transduction pathway where it activates this transcription factor Hi5 in the nucleus, which in turn will bind to the promoter and regulate the expression of several genes involved with photoprotection, DNA repair, and this will give ultimately the UV protection, the acclimation response of the plant towards UV, and the repair of UV damage. Also known is some negative factors in the pathway. These are these RUP proteins, RUP1 and RUP2, which are negative players. And one of these BBX factors, which Arpita was working with, one of them has also been previously identified as a negative regulator, BBX24, in this UV signaling pathway. So let me tell you a bit about these BBX proteins. So these BBX proteins, as I told you, the name BBX comes from that they have this domains known as B-box domains, which are nothing but they are domains which are rich in cysteine and histidine. Here you have a consensus sequence of the B-box domain. In Arabidopsis, the model plant with which we are working, there are 32 BBX proteins, and these 32 BBX proteins are divided into 5 structural groups, group 1 to group 5. The protein which Arpita was working with was BBX31, which belongs to this group 5. And here you see that the BBX proteins can have different structural motifs in addition to the B-box, which are shown in blue. You have the VP motif and the CCT domain. So depending on the presence and absence of these domains, they have been grouped into these structural groups. And BBX31 is the one which has only one B-box in its N-terminal end. Now, before we started looking at the function of BBX31 in UV production, what we thought was, is there anything in the literature which suggests that BBX31 might have some role to play in UV signaling? So we got hold of a very old paper back from, I think, 2004 in PNAS, which involved some of the world leaders in the UV signaling field in plants. Roman Ulm is one of them. So in that paper, that was the first time they had identified the role of Hi5 in UVB signaling, the transcription factor Hi5, which is a major player in the UV signaling pathway. And among the targets of Hi5, they had found several genes which are involved in transcriptional regulation. And this is a list from the microarray they had done. And under the transcriptional regulation head, you see that the first one, which was identified, was Constance B-box zinc family protein. Okay, and this turns out to be BBX31. Okay, so it was hidden somewhere in the microarray data almost 15-16 years ago, but somehow no one had worked on this before about the role of this gene or protein in UV protection. Another quite recent paper, from another very well-known group, Gareth Jenkins group in Glasgow. So they worked on the UV receptor UVR8, which also acts as a chromatin modifier, and they showed that among the targets of UVR8, they had again identified, this was again a high throughput study, and they had identified several, and in that list, one of them was BBX31. So this gave us some confidence that probably we are on the right track. There is some evidence which suggests that BBX31 might be playing some role in UV protection. So the questions we asked was, what is the role of BBX31 in light signaling in general, first of all? And the second, how does BBX31 provide tolerance to UVB? So to answer this, the first thing we did was we checked the expression of this gene BBX31 under UV treatment. So the promoter of BBX31 has a UV box, which is present in several genes which are involved in the UV signaling pathway. So this was another suggestion that probably this gene has something to do with UV pathway. When we looked at the expression of BBX31, what you see here in panel B, figure B, is at different time points we looked at, and this is time after the UVB treatment, and the gray bar is under white light and the black bar is under UVB. And what you see is that under UVB there is increased expression of BBX31. Approximately after 1 hour after UV treatment, its expression peaks. We also checked how the expression is related with the dose of UV. So we gave it different doses, different strengths of UV, UV1, 2, 3. These are increased doses of UV. And we see that as you increase the dose of UV, the expression of BBX31 increases. In panel D, what you see here is Col0 is the wild type plants. And here in black is the expression levels in white light, and gray is expression levels under UVB. So you see that when you give it UVB, the expression of this gene increases in the wild type plants. I showed you the pathway of UV signaling, where UVR8 is the receptor, followed by COP1 and HY5, which are major players in the UV signaling pathway. And what you see is that if you mutate these genes, then the increased expression that you see under UVB in the wild type plants, you lose that increased expression, indicating that this BBX31 is probably playing its role in the canonical UV signaling pathway. So if you mutate any of these genes, you don't get that increased expression. Now, HY5 is the most downstream target in that UV signaling pathway, and we saw that that the expression of BBX31 is under regulation of HY5. So we looked at the promoter of HY5, and we found that it contains a G-box. G-box is one of those elements in the promoters where light-regulated elements where HY5 is known to bind. Okay, so it's a 6 nucleotide CSE GTG this motif. So we wondered whether HY5 is able to bind to this G-box to regulate the expression of BBX31. So here we did an AMSA assay, and we could see. So what you see in this panel A is the AMSA where we checked the binding of HY5 to the promoter region of BBX31, and we also made mutations in the G-box. So wherever you see plus is where you have the presence of either the promoter region of BBX31 or the mutated version of BBX31, and then the HY5 protein tagged with GST added together with the controls. And what you see here is that HY5 can bind to the promoter region of BBX31, which contains the G box. But when you mutate the promoter, the mutation is in the G box, then you cannot see the band is missing, indicating that Hi5 is unable to bind to BBX31, suggesting that Hi5 binds to this G box. We also did some chip assay to check the enrichment of Hi5 on the promoter of BBX31, and this chip followed by qPCR is what is shown in panel B. And here you see that col0, which is the wild type plant, and Hi5 mutant, which was used as a control. So in col0, what you see here is that the black bars indicate it's the like the IgG antibody control, and the gray bar indicates the Hi5 when probed with the Hi5 antibody. So what you see is that there was enrichment of the Hi5 in this col0 line. And what is interesting to see here is that this enrichment of Hi5 on the promoter of BBX31 was irrespective of whether you treat the plants with UV or without UV. So you see minus UV and plus UV. In both conditions, there was an enrichment of Hi5. So this indicates that Hi5 can constitutively bind to the BBX31 promoter. Okay. But however, when we looked at the expression, binding is constitutive. It can bind whether UV is present or not. But when we looked at the expression, then we see that this expression is very specific to UV. So what you see here is in col0, the black is white light and the gray is UV. Under UV, the expression increases. If you overexpress Hi5, then there is even enhanced expression of BBX31. So this suggests that the Hi5 is constitutively binding to 31 promoter, and it enhances the transcriptional levels, specifically under UVB. Now, to understand better what role BBX31 plays, we ordered some mutants of BBX31. First, we ordered them from the stock center, but unfortunately they did not have reduced expression levels of BBX31 in those lines. So then from our collaborator, we got some microRNA gene-induced silencing lines and CRISPR lines, this MIP and BBX31-1 respectively. And we started studying them to understand what role this gene plays in the light signaling and as well as the UV signaling. So what you're seeing here, because this is the first time I'm showing you real Arabidopsis seedling. So what you're seeing here are Arabidopsis seedlings. So these are very young, 6 to 7 day old seedlings. And the green part are the cotyledons. The stem-like part is the hypocotyle. And light has a role that once a seed germinates and the hypocotyle elongates and then the cotyledon expands and opens and turns green. So light will try to prevent the elongation of the hypocotyle. This is a property which is I'll discuss further in the second part of the story. I'll discuss it in more detail. So photomorphogenesis, so the light-dependent morphology. So light tends to make the hypocotyle shorter or inhibit the expansion or elongation of the hypocotyle. So if you have a gene which when you mutate, if you have shorter hypocotyles, then that would indicate that the loss of that gene made the hypocotyle more, the plant more photomorphogenic. The phenotype was more photomorphogenic, indicating that it's a negative regulator of photomorphogenesis. Okay, and the vice versa. So what you see here, if you look, you have different genotypes. col0 is the wild type. Then you have the mutants MIP1A and BBX31, and then you have the overexpressor lines. 35 has BBX31, 2 lines of the overexpressor. Number 1 and number 2, and below that what is mentioned is in the absence or presence of UVB. Okay, so first let us look at only in the minus seedlings, only in the absence of UVB. That means that it is in white light conditions. And what you see here is that the hypocotyl length in the this is the control call 0, and when you have mutated this BBX31, the MIP1A and BBX31-1, then you have shorter hypocotyls compared to the wild type. And if you overexpress them, you have longer hypocotyls. Okay, they are taller, the seedlings are taller. So this indicates that it's a negative regulator of the white light photomorphogenesis. Now, in UV light has a property that it will further reduce the hypocotyl length. So you can see in col 0 seedlings, which is the control, once you give plus UV, the hypocotyl length is shorter compared to without UV, minus UV. This trend is followed in all the genotypes. You can see that it always gets shorter under UV. But the amount, the percentage of reduction that you see is lesser in the mutants. You see that the amount of reduction in MIP1A and BBX31-1 is comparatively lesser than what you see in the col 0 seedlings. On the other hand, if you look at the overexpressors, the percentage of reduction is higher. So this indicates that it is a positive regulator of UVB signaling. So here we found there was some difference in the way it is acting in white light and the way it is acting under UVB. Now, because we had previously seen that it is in the canonical UV signaling pathway, and Hi5 binds to its promoter. So we wanted to know how Hi5 regulates its role in photomorphogenesis. So for that, what we did, this is again those Arabidopsis seedlings, different genotypes which are indicated below, plus and minus indicating presence or absence of UVB. And here you see that in addition to col 0 and BBX31, we included another genotype, Hi5. This is the transcription factor, which is like a key modulator of light signaling, and this is the one which was binding to the promoter of BBX31. So Hi5 is a positive regulator of light signaling, meaning if you mutate it, then it produces really tall seedlings, so long hypocotyl. And so what you must appreciate here is the negative regulator BBX31. When you're overexpressing it, you're getting tall seedlings, and the positive regulator, when you're mutating it, you're getting tall seedlings. Now, when we made double mutants of Hi5 with BBX31, we make crosses between these different lines, and we also overexpress BBX31 in Hi5 mutant background. Okay, and what you will be able to see here is when we made the double mutants, the hypocotyl length, both in white light as well as the UVB, if you look only at the wild type for the first instance, then you see that the hypocotyl length is intermediate between BBX31 and Hi5. Similarly, when you have overexpressed BBX31 in Hi5, then you get some super tall seedlings. Okay, so meaning that when you and that is what has been shown in this graph below, that if you look at there, then BBX31 has shorter than wild type, Hi5 has taller seedlings than hypocotyl than wild type, and when you we generate a double mutant, it is intermediate. Hi5 mutant has tall, overexpressor of 31 has tall. When you combine these two, you get super tall. So what this suggests is that these two genes are doing things independent of each other. Okay, that is why when you have the double mutant, it is intermediate. When you have the mutant of one and the overexpressor of other, you have even stronger phenotype, even taller seedlings. This was also confirmed when we did a RNA-seq experiment where we looked at genes which are being regulated by Hi5 and 31. And what you see here is that there was very less overlap. There are only 7 genes which overlap between Hi5 mutant, which is the tall mutant, and the 35S BBX31, which is another tall mutant. So in both of these, there was very less overlap, suggesting that the genes these two transcription factors are regulating are probably different, and they're acting independent of each other, at least in this control of photomorphogenesis, the hypocotyl elongation. So both in upregulated and downregulated, what you see is there was very less overlap between the two genotypes, the genes regulated by the two genotypes. Now, we went back to the question with which the project started, which was about UV protection. So here, as I had shown you in the first slide, what was happening was in panel A, what you see is that if you mutate BBX31, then they bleach more than the wild type. And in panel B, what you see is that if you generate lines which are overexpressing BBX31, we had 3 different independent lines here, then they stay more green under UVB compared to the wild type called 0. Okay, we measured the chlorophyll content in these lines, and what we saw was that it was lesser in the mutant and it was higher in the overexpressor. We also measured anthocyanin. These are all factors which contribute to protection, to photoprotection. And here again, we found that there was some lower amounts of anthocyanin in the mutants and slightly higher in the overexpressor. And together with the help of our collaborator in IIT Mandi, we did some metabolomic analysis, and there we identified some phenolic compounds which were found to be upregulated specifically in this overexpressor lines. These were this polyphenol comeric acid, gallic acid, hydroxybenzoic acid, and vanillic acid. So these are those compounds which are acting as the photoprotectants and providing this enhanced protection to these 35S BBX31 seedlings so that they can remain green under UVB for a longer time. Now, this protection factor, we wondered that whether Hi5, which in the photomorphogenesis we found that these 2 genes are acting independently, but we asked the question that what happens in this UV protection, the UV tolerance phenotype that we see in this one, how are these 2 genes interacting? So here, what you see in panel A is the 4 quarters, the col 0 is the wild type, and you have the Hi5 mutant, you have the 35S overexpressor, and you have the overexpressor in Hi5 mutant background. This is in white light condition. Once you move them to UV, you see the UC start in the first signs of bleaching in the wild type. In Hi5, you see enhanced because it's a positive factor in the UV signaling pathway. So if you mutate it, plants become more susceptible to UV. In the 31 overexpressor, they are more resistant, they remain more green. But if you overexpress 31 in the Hi5 mutant background, even the overexpression of 31 is not helping the plants. They still become susceptible, they are more susceptible to UVB. This indicates that that 31 is promoting UV tolerance in a Hi5 dependent manner. It requires Hi5 for this protection. Okay, so this was also done in terms of, we did the measurements of chlorophyll in these different lines, and it fell in line with the phenotypes that we saw, that in Hi5 you have lower chlorophyll levels, and in BBX31 you have higher, but if you overexpress 31 in Hi5 mutant, again it is similar to Hi5, it's lower. Also, the genes which are being regulated by these factors to provide UV protection, these involve these ELIP genes. These are early light induced proteins. You have the genes involved in the anthocyanin biosynthesis, chalcone synthase, chalcone isomerase. UVR2 is a gene involved in DNA repair. So all these genes get upregulated when you overexpress 31. That is what is shown in this figure E. You see that they are highly upregulated in the 31 overexpressor. But if you overexpress 31 in Hi5 mutant background, again you see that they come down. So this all the genetic analysis, the physiological parameters that we measured, the biochemical analysis, everything indicated that the UVB protection function of BBX31 is Hi5 dependent. Now, Hi5 dependent means that it is acting through Hi5, so Hi5 must be downstream of BBX31 in this pathway. So BBX31 is also a transcription factor. So we wondered that initially we had seen that Hi5 can regulate BBX31 expression levels. So we wondered in this pathway, in the stress regulation pathway, is it possible that 31 is regulating Hi5 because it is acting downstream of Hi5 in this pathway, downstream of 31. So in panel A, this is in white light condition. Here, when we checked the expression of Hi5, then we did not see much difference in the mutants of 31. But under UVB, you see that when you overexpress 31, there is almost 10-fold increase in the expression of Hi5. Okay? Although in the mutant, we could not see a significant difference. So what this suggests that there might be some regulation under UV conditions where 35 is regulating the expression of Hi5, especially particularly when you overexpress BBX31, the Hi5 transcript levels, they go up. And this upregulation, which you see in panel C, if you look, this upregulation, which has been shown in gray bar under col 0, this can be abrogated if you mutate UVR8, which is the photoreceptor for UVB. So when you mutate this, you no longer see this increased expression of Hi5. And even in this line, when you have the mutant of when you have the UVR8 mutant and you have overexpressed 31, you don't see this increased expression of Hi5, indicating that that this is in a UVR8 dependent fashion. So taking together, what I tried to tell you in this first story was that we identified a factor called BBX31, which under white light acts as a negative regulator of photomorphogenesis. And under UV light, it is acting together with Hi5. It's making some reciprocal interactions with Hi5, where Hi5 and 31, they independently regulate the UV-mediated photomorphogenesis. Hi5 can regulate the transcriptional levels of BBX31 by binding to its promoter. 31, when you overexpress 31, then there is enhanced Hi5 transcript levels. And in a Hi5 dependent manner, BBX31 is regulating the UVB-mediated stress response. Okay, so this was Arpita's work, which was published last year. And an article about it was also written in Hindustan Times last year, where the importance of this work was discussed. Okay, so now let me move to the second story. This is the more recent story, the story that we managed to finish during the lockdown, but it published during the lockdown period. But that is not why I am calling it the lockdown story. I'm calling it the lockdown story because the story is about how plants mediate lockdown under stress. Okay, so what I mean here is that normally this is in a very layman's fashion, this has been made, this figure. So sprouting meaning the germination. So everyone knows, probably all of you know about this germination. So once the seed has germinated, after that, it has to take a very important decision that whether it should start the greening process, open up its cotyledons and become autotrophic. Okay, the basic feature of all plants. So this decision making is very critical because it involves a lot of energy, to invest a lot of energy in this growth process. And if the conditions are not favorable, if there is stress, either whatever, salinity, drought, whatever, if there are stressful conditions, then the plant would prefer not to invest energy in growth because ultimately it will die. It will try to remain in a quiescent state until there are favorable situations. Okay, so this step after germination, after the seed coat has opened up, the radical has come out, this step is known as the seedling emergence and establishment. Okay, where the seedling starts the greening process and opens up. So here, depending on the stress conditions, the plant can enforce a lockdown or a arrest, a developmental growth arrest. Okay, so how this is done is there's a hormone which was discovered almost 80 years back called abscisic acid. Okay, and it was shown that this hormone plays a very important role in the early developmental steps of the plant. So starting with the rice seed, once it is imbibed, it has water, imbibition will happen, the testa will rupture, and the radical will emerge. This step is the first step, which is known as the germination. This step is inhibited by this hormone known as abscisic acid. Okay? And after the radical emergence, there will be this hypoglottile emergence and then the seedling establishment. All these steps are known as the post-germinative developmental phases or the post-germinative growth. And here also ABA, abscisic acid, plays an important inhibitory role, that it will inhibit these steps from happening. Now, this hormone is known as the stress hormone. So it will allow the plant to cope up with stress, not invest too much energy in in growing, so that it can cope up with the stress. Now, how this ABA understands whether the situation outside is favorable or not, ABA understand how the plant understands includes some crosstalk between external factors and the levels or activity of this hormone ABA in the seed. Okay, so for germination in particular, it is quite well known that light plays a very important role in this crosstalk or interplay. So the external cue light will inhibit the role of ABA in inhibiting this germination process. So what that means is light promotes germination and the hormone ABA inhibits germination. So there is a antagonistic relationship between light and ABA in this germination process. Now, what happens during post-germinative growth was not very well known. Okay, so there was some knowledge gap that how does the external cues, especially light, what is its role in regulating this process, where we also know that ABA plays an important role in inhibiting. So the question we asked was that what is the effect of light on ABA mediated hello? Hello?
Sourav?
Can you still see the slides?
Yeah.
Okay. I just one moment. Oops. Yeah. So what is the effect of light on this ABA-mediated post-germinative seedling development? This is the question that we asked. So before I go into the details, I'll just like to explain some of the nomenclature so that it's easy to understand the experiment. So what I'm showing you here in the initial figure is one is a cartoon and one are the actual Arabidopsis seedlings when they are grown in dark and in light. And what you see is that there is a tremendous difference between the morphology of seedlings depending on whether they are growing in light or darkness. So in darkness, the kind of development is called photomorphogenic growth, where you have a very elongated hypocotyle. So this part is the hypocotyle, and the cotyledons are closed. They are folded cotyledons. They form an apical hook. The reason for this is that the cotyledons are hiding the shoot apical meristem and protecting it from the soil and particles and everything abrasive material, so that once it comes out of the soil, then from the shoot apical meristem, all the adult plants of the parts of the plant can develop. Sorry. The other development is the photomorphogenic development, which is when the seedling sees light. Here what you see, as I told you before, the hypocotyle expansion is inhibited. So you have short hypocotyle. The cotyledons, they open up. They are green in color. And of course, now the shoot apical meristem is exposed. And from here, you will have the internode, the true leaves that you see that, and the adult parts of the plant will start developing. Ultimately, these cotyledons will, once the seedling has reached the surface of the light, ultimately these cotyledons will be shed off. And then the adult growth continues. The dart dependent growth is something which you have to imagine as when the seed was buried under the soil. So it was trying to come out to the surface. That's why it was elongating its hypocotyle. Now, this is the kind of development you see in light and dark. Now coming to this experiment with ABA. So here, this work was done by another very talented PhD student, Yadu Krishnan, in the lab, who recently finished his PhD. So Yadu, he put these Arabidopsis seeds in light and in darkness under different concentrations of ABA. As I told you, ABA inhibits the process of both germination and the post-germinative growth, and that is what you see here. So 0.5 and 1 micromolar ABA is the increasing concentrations of ABA. So under light, you see that there is enhanced sensitivity towards ABA. The plants are showing less of this, the greening that you see here, the cotyledons opening and greening, that is lesser as you increase ABA. But what is important to see here is in darkness, this sensitivity is enhanced. Okay, so there is an enhanced sensitivity towards ABA. Even in 0.5 micromolar ABA, you can hardly see any of these cotyledons coming out. So this is what is being shown in the graph here. So in red line you have the seedling establishment in light, and in black line you have the seedling establishment in darkness. What you can see here is by establishment, what we mean is that the cotyledons have opened up completely, and the hypocotyle has emerged, the cotyledons have opened up, and they have started greening. So the seedling establishment, when Yadu measured, then he saw that when there is no ABA, you see that the black and the red bar, the red line, they're almost similar. So similar establishment rates in absence of ABA. But when you increase the concentration of ABA, you start seeing a big difference between whether the seedlings were grown under light or in darkness. Okay, so seedling establishment is much lesser when it is grown in darkness. So that means that the sensitivity towards ABA is much higher when the seedlings are being grown in dark. So now this told us that probably there is some factor which works in darkness, which is regulating this sensitivity towards ABA. And COP1, constitutively photomorphogenic 1, was identified back in the 90s in a genetic screen, where it was identified as a protein or gene which is very active in darkness. And what it does is that it prevents photomorphogenesis to happen in darkness. What I mean here is in the figure, what you're seeing here is wild type seedlings grown in dark and in light. And this I explained to you before, how it looks under dark, the elongated hypocotyle, closed cotyledons, apical hook, and in darkness, short hypocotyle, open and green cotyledons. So this is the normal phenotype that you will see in wild type. But the genetic screens identified a mutant COP1, which even in the dark, it looked like as if it is seeing light, it is growing under light. So even in the dark, it had short hypocotyle, it opened up its cotyledons, although the greening was not as much as you see in light. But several of the phenotypes were similar to what you see in light. That's why the name of the mutant, constitutively photomorphogenic. It was always looking as if it is a photomorphogenic phenotype was seen. So this COP1, this is a protein which is very active in darkness. Light inactivates this protein, okay? And the function of COP1 is that it inactivates photomorphogenesis, specifically in darkness. Okay? So we wondered, and this is the protein structure of COP1. So it has this ring domain, coil domain, WD40 domain, which is a 675 amino acid protein. So we wondered whether this protein, which is known to be very active in darkness, if it has any role to play. So before we go into that, just to tell about the molecular function of this protein. So COP1 is a E3 ubiquitin ligase. So what it does is it will ubiquitinate different proteins, and the degradation through the 26S proteasome is mediated by COP1. And as I was telling you, that it is a protein which is very active in darkness. So in dark, COP1 is nuclear, and in light, its activity is reduced both by its nucleocytoplasmic partitioning, as well as several other factors about some other proteins like SPA proteins. It makes complexes with them. So these all complexes are inhibited during light by several other factors. I'm not going to the details. So its activity is reduced during light. So this kind of fit well with what we were looking for, a candidate which is very active in darkness. So here is the experiment that was done. This is what was shown to you before, that what happens in normal wild type seedlings. And this is what happens in COP1 mutant. So in COP1 mutant, if you see the similar experiment which was done before, when this one was done in COP1 mutant, if you look at the dark seedlings, you see that there was very strong inhibition in the dark in the wild type. But when you have a COP1 mutant, you see that there is not a very strong inhibition in seedling establishment in the dark in the COP1 mutant. Meaning that when you have inactivated this gene, or when this gene is absent, then this enhanced sensitivity is in darkness. So this suggests that probably COP1 is important in this enhanced sensitivity towards darkness. So this is what has been measured here. You see that there is a big difference between the red line and the black line in the case of the wild type seedlings, which is the light and the dark seedling establishment. But when you have a COP1 mutant, you see that the difference between the light and the dark seedling establishment is very less. Okay, so the ABA-induced inhibition of this post-germinative seedling growth seems to be COP1 dependent. Now, this COP1 is a protein which is quite an ancient protein, in fact. Actually, you have COP1 both in you have it before the divergence of the plants and the animals. Okay, there is COP1 protein even in animals, where it plays role in the tumor suppressor pathway. So here, let us discuss about the plants. So in plants, this is like just showing you the evolution of the land plants once they moved from aquatic environment towards land. And what it looks like that this COP1 is quite ubiquitously present in different plant species, right from so like even the green algae, the mosses, the lycophytes, ferns, gymnosperms, so all of them have this COP1. The ABA pathway probably came into existence during this transition from water to land. So as I told you, ABA is a stress hormone, and plants, this hormone came into existence for the plants to conquer the land. When they moved from aquatic environment to land, there was a lot of stress involved, and to cope up with this stress, the evolution of the ABA pathway probably started somewhere at this point. Now, what we tried to do here was we looked at how conserved is this function of COP1 in the ABA pathway, and for that we looked at COP1 present in a moss, a very ancient plant, Fiscometrella patens. And we got a line from one of our collaborator, Ute Hooker, where the Fiscometrella patens COP1 was overexpressed in the Arabidopsis COP1 mutant background. Okay, so the idea was to see whether the functions of this COP1 in this dicot and in this mosses, are they conserved? And here is the experiment. And what you see here, I'll just try to give you the final message here, is if you look at the plus ABA line, so here you see that COP1-4, it shows it is hyposensitive to ABA, so they are germinating and opening up their seedlings. But when you are overexpressing the Fiscometrella patens COP1 in COP1 mutant background, then you again see this enhanced sensitivity towards ABA, indicating that the Fiscometrella COP1 was able to rescue the phenotype, the COP1 mutant phenotype. So so that says that this function of COP1 in the ABA pathway is quite evolutionarily conserved. Now, after having done this, the next question that we asked was how does COP1 modulate the ABA signaling pathway? Okay, so we understood that COP1 is doing some role. COP1 was initially always characterized with its role in photomorphogenesis. Here we got some clue that it is playing a role in the ABA signaling pathway. Now, how is COP1 modulating this hormone pathway to regulate the germination, post-germination development is the question we asked. And here the first thing that we tried to do was to look at evidences of which are the major players in the ABA signaling pathway. And ABI5, it's a transcription factor, which has been shown to play important role in this post-germination developmental arrest. And here you see its phenotype under ABA in this WS is the wild type and ABI5 mutant. Even 5 days under 5 micromolar ABA, you see that the seedling can open up its cotyledons and it shows post-germinative development. So we asked whether there is a relation between COP1 and ABI5 in regulation of the ABA pathway. And here what we did was we overexpressed ABI5 in the COP1 mutant background. So you see initially COL0, COP1, the overexpression of ABI5 and in the COP1 mutant background. What you see here is that the overexpressor, when we overexpress the ABI5 in the COP1 mutant background, then we see you can rescue you have open cotyledons, and there is the post-germinative development is happening here. So this is what has been shown in the graph below. Okay, so this told us that COP1 is acting downstream to ABA, because the phenotype of the 35S ABI5 in COP1 background was similar to COP1. So this indicates that COP1 is acting downstream to ABI5 to promote the ABA-mediated post-germinative growth arrest. So now we looked at, initially we had checked about the expression level and both at the transcript level and the protein level, but we did not find any differences. Ultimately, what we found was that the downstream genes to which ABI5, I told you, the transcription factor, it is regulating several genes either directly or indirectly to control this post-germinative development. And our results indicated that what COP1 is actually doing is it is modulating the binding of ABI5 to its downstream genes. Okay, so here what I am showing you, EM1 and EM6 are two of these direct targets of ABI5, and the motifs shown in orange are the G-box type ABRE, which are the ABA response elements, and we could see there was enrichment of ABI5 on enrichment of ABI5 on the EM1 and EM6 promoter regions. So that is what is the chip followed by qPCR, which has been shown here below. So you see that if you mutate COP1, then you see that this enrichment of EM1 of the ABI5 on the EM1 and EM6 promoter goes down, indicating that that the promoter binding and of ABI5 on its target genes is being modulated by COP1, and it will be reduced if you mutate COP1. So basically, what we found here was, I started with this question, that the question we asked was that how light is integrating with this ABA pathway to regulate this post-germinative development. There was a lapse in the knowledge here, and what Yadu's study has shown now is that the molecular factor which is connecting these two pathways, the light pathway and the ABA pathway, is COP1. So light inhibits COP1, and COP1 plays a positive role in the ABA signaling pathway, where it promotes the binding of ABI5 to the promoter of the target genes. So here we have found an interplay between light and ABA in regulating post-germinative growth arrest. This work was recently published in Plant Journal. And so in a nutshell, what this story was about was that when there is a factor called COP1, which is a major player in light signaling pathway. It is a factor which is very active in the dark. When there is light, the activity of COP1 goes down because of its nucleocytoplasmic partitioning and other regulations. And this factor COP1 in the light signaling pathway integrates with the ABA signaling pathway, where it is actually modulating the binding of ABI5, which is a key modulator, key player in the ABA signaling pathway, to downstream genes to regulate this process of post-germinative development. So that is what is shown here, that in darkness you have lots of COP1, and because it is positively regulating ABI5 binding to the downstream genes, so the process will be, post-germinative development will be inhibited in darkness. But when you move to light, the activity of COP1 decreases, and the regulation of ABI5 will be reduced, and that's why the plant will move towards post-germinative development. It will open up. Ecologically, if you think, it is like the resource allocation. Once the conditions are conducive, there is lots of light available, then the plant thinks that now it is time to invest energy in growth rather than in stress tolerance. So what it is trying to do is through light, giving a cue through COP1. So as soon as light comes, COP1 switches off, and when COP1 switches off, the ABA levels or the ABA activity in the plant goes down, and that leads to the post-germinative development in the plant. So this is about the second story which I wanted to tell today. Some of the other works which are going on in the lab with respect to plant-environment interactions, which I could not talk today, was about some of these BBX proteins. Some work with the nutrient, there are some mate factors which we study and how they regulate aluminum tolerance and sensitivity. So these are some of the other regions where we have been working, but maybe some other time I can tell you about it. This is the group. I apologize, the last member, Ajar, was not in this image, as this image was taken during Arpita's defense last year. But I think other than him, almost everyone is in this image. So yeah, I especially would, the work that I talked today, primarily the first work was Arpita Yadav's PhD work, and the second work was Yadu Krishnan's PhD work, and it was also, they were helped by other members in the lab. I would like to thank IISER Bhopal, DST and DBT for funding, and our collaborators Shyam Masakapalli at IIT Mandi, Stephen Wenkel at University of Denmark, and Henrik Johansson in Germany. With this, I would like to end. I am not sure if I overshoot the time. Yeah, so sometimes it might be good to forget about your experiment. If yeah, that's how our first story began, and we ended up finding something interesting. I'll be happy to take some questions.
Thank you, Sourav. Thank you. Thank you for giving an excellent presentation. Now it's time for discussion. Now we have some. Hello?
Yeah, hello. Yes.
Yeah. Now we have some question.
Yeah.
Yeah. First question is from Ishida Chatterjee. Dog1 gene acts as the switch between germination and dormancy. Is it related to COP1 in any way? Is the question.
Yeah. So so the part which I discussed today, so dog1 is very important in the dormancy, as Ishita mentioned, dormancy and the germination processes. So Wim Soppe, I think he is the leader in this. He probably discovered dog1. So in those aspects, the role of COP1 is yet to be like we don't know yet. But what we found was specifically in this post germination development, COP1 plays a very important role.
Hello?
Yes.
Yeah. Now we have second question. Hello?
Yes.
Can you hear me?
Yes.
Yeah. It's a doubt. Normal physiological metabolism of plants or crop might be tricky as AT genome is fully known, but other crops may be different. It's a doubt. It's clear for you?
Can you just repeat it once more?
Yeah, sure.
So normal physiology?
A normal physiological metabolism of plants or crop might be tricky as AT genome is fully known, but other crops may be different.
So definitely. So I agree that this is so the idea is to start with a model organism, get some clues about what is happening in Arabidopsis, and then now what we are trying to do is to move ahead and to see are similar things happening in crop plants. Okay, so that is the next step that we are taking now. Of course, having said that, although I said that the COP1 is very ubiquitous and it is found in almost all plants, but COP1 is a very it's kind of too important. Okay, if you play around with COP1, there are a lot of things which can happen. So that's why it's not very easy to very specifically modulate this process, because as soon as you you modulate the levels of COP1 by some means, it can have some other effects as well. So to specifically understand what it is doing in this pathway, we have to probably identify some other factors which are giving COP1 the specificity to act in post-germinative development. Once we know that, then probably doing some work for crop improvement in the future might be possible. So this is mostly related with, it will be related with crop emergence and establishment, okay, how this knowledge can be used.
I think it's clear. Next question is, how do we translate these findings on model plants into plants from natural habitats?
Yeah, so I think I kind of partly answered that question in the previous, and the previous question also was kind of, yeah, hello?
Yeah.
I think I partly answered that question in the previous question. Previous question was kind of similar. So as I said, these are early days. So we have got some lead, and we have started working on like this. This is in the pipeline.
Okay. Now we have no more questions. Thank you, Sourav.
Okay, then thanks a lot. It was really nice. And if there are any more queries, feel free to send me an email. I will be happy to answer.
Okay, sure. Thank you.
Thank you.
Yeah.